CAR T-cell therapy uses the patient's own immune cells, which are modified to attack cancer. Specifically, we work with T cells, also called T lymphocytes, which naturally recognize virus-infected or cancerous cells. These T cells are genetically modified to add a special receptor called CAR, which allows them to recognize and bind to specific proteins present in cancer cells. When the CAR receptor recognizes these specific proteins, generally called antigens, the CAR T cell exerts an anti-tumor activity against the cancer cell. Each type of cancer is different, and cancer cells of one type or another present different antigens. Therefore, for CAR T therapy to be effective, it is crucial to develop a CAR receptor that can recognize the appropriate antigen for that specific type of cancer. For example, RE1 is a CAR T therapy developed against the CD19 antigen, which is present in acute lymphoblastic leukemia, while RE2 is a CAR T therapy developed against BCMA, an antigen of interest in multiple myeloma. Given their positive results, both therapies are currently approved for clinical use in Spain. As a mechanism of resistance to treatment, some tumors lose the target antigen, reducing the therapy's effectiveness. For this reason, IDIBAPS and Hospital Clínic of Barcelona have developed RE3, a new dual CAR T that recognizes two antigens, CD19 and BCMA, both of which are co-expressed in non-Hodgkin lymphomas. During its development, the team co-led by Dr. Sonia Gadon and Alvaro Urbano studied different strategies and methodologies to produce dual CAR T cells, including using a mixture of the two individual CAR T cells working together. Generating bicistronic CAR T cells expressing both CAR receptors, producing CAR T cells presenting both CAR receptors in tandem, and the most effective approach, co-transduction of two viral vectors. In this case, the modified T cells displayed a variety of CAR receptor expression profiles. Some CAR T cells expressed only the anti-CD19 receptor, others expressed only the anti-BCMA CAR, and between 15 and 20% of the cells exhibited dual expression of both anti-CD19 and anti-BCMA CARs. This result is what we now call RE3. Studies conducted with patient biopsies and murine models showed an improved anti-tumor effect of the dual CAR T RE3 developed through this co-transduction process compared to other strategies. And in particular, compared to the individual CAR T therapies RE1 and RE2, demonstrating greater anti-tumor efficacy, survival, and safety. Thanks to these preclinical results and the previous experience of IDIBAPS and Hospital Clínic of Barcelona with CAR T cell therapy, RE3 is currently being studied in a clinical trial for patients with non-Hodgkin lymphoma.